PowerPoint because I just want bare minimum. For example, what is genomics? Okay,
Chapter 9.
so what is? Remember, we use the word annotation. So what is genome, genomics, and then we talked about annotation. Where is annotation? So here the word annotating genes. What's genome assembly annotation? Okay, this is Sanger's method. You should know Sanger's method. Okay. Now there are first generation, second generation. I'm sorry, I'll be out of here in 5 minutes, okay? I'm so sorry. So here, okay, now there are, I'm not going to do this. We don't want any of this. But anytime evolution, we talked about horizontal gene transfer. See, horizontal gene transfer is everywhere. And what's the mobilome? You should know what the mobilome is, okay? And then we also talked about this will be just definitions. So here will be we've repeated this so many times, right?
No.
All right, so these chromosomal islands are the ones that will insert, and pathogenicity islands are those that, so here in E. coli, if you see the normal genome, it's smaller. But if you see the pathogenic, it's bigger because it has those segments that have inserted themselves either by transformation or by transduction or by conjugation. Either way, okay. What is the word metagenomics? What does it mean? So here, you need to just know the definitions, all right? So this would be a good idea. And how do you do this? The microarray. This is a technique, not in detail. And then we also did this was the phylochips, which is a type of microarray. But the advantage of phylochip is that the phyllo means phylogenetic. So you can find out the species. Like for me, I remember, my God, I had found 4 new species, and it took me forever to find what they belong to, because until I don't know, I will not be able to find any of the enzymes, right? These are extremophiles. So now is the RNA genome sequence analysis. You can look at that. Now, what is proteomics? Proteomics, what is interactome? These are just definitions. So just DEF, that means definitions that I'm asking you. Definitions, okay? So find out the definitions, okay. Now, not the method. What is the interactome? Interactome, obviously, what is this interactions? Metabolomics. Now, metagenomics, metabolomics. Obviously, it comes from the word metabolic, right? So here, all the metabolites. It's important because, remember, we learned one method, right? Like glycolysis or Krebs cycle. But we never learned, what if there are so many different conditions? The conditions that are present in your body right now may not be in his body or my body. We are different, right? Because different genes, different food type, different lifestyle. So many things. So this will study an entire play of this. And they talked about something of doing in a permafrost, which you can look at. Not important. Single cell genomics is coming up right now because I need to find out what about me. I don't want to talk about the entire population. I'm talking about me. I'm sure you must have heard okay, I don't know all these people. Jennifer Lopez, is she the one? J-Lo? Mm-hmm. Okay, J-Lo is the one who, she got both her breasts removed. Is she the one?
No, that was Angelina Jolie.
Okay, Angelina Jolie. So she found that she had the defective gene, okay? Mm-hmm. All right, so there you go. That's single cell genomics for you. So single cell genomics is the one, and right now this is the new trend. Like you have a child and they want to find out what's the % chance or this. So here, this would be interesting because that's what they found for tuberculosis, all right? This is they are using this, and they are using for like breast cancer, tumor growth. They found out about 2 genes. That's all. Only definitions. There'll be few questions. Now, chapter 10 is going to be in detail. All right. So chapter 10 is the Baltimore. Baltimore is the most important. I'm going to put like 1, 2, 3, 4, 5, 6. That's how important it is. You should be able to tell me all the classes. Okay. Yes, and examples. No, it's easy to get confused. Yeah, that's what I'm saying.
Maybe
so which class do you say, for example, class 5? Okay, so class 5, I know it is the negative, single-stranded RNA strand. Example is a flu. It is implanted in my brain. Do you understand? I cannot forget it.
Is recombinant.
Okay, with retrovirus. And 7 is partially double-stranded DNA, which uses retrovirus. You see what I'm saying? So you should know this that well, okay, for long answer question. Very important. All right, so here, even if you do, I'm not interested in this. Now, we're talking about DNA virus. And remember, I did not choose this. I chose bacteriophage M13. So you should know bacteriophage M13, why it is used for cloning. All right. Then Mu. What Mu? Why did I want to know about Mu? Remember, it has transposons, and it uses the enzyme transposase. Anytime you have transposons, that means you are going to cause genetic mutations. Okay? So then, of course, you have to have DNA. The archaeal viruses are all DNA. They all are double-stranded. We don't know of any RNA archaeal viruses. There are still studies going on. They're thinking they have found, but nothing is sure. Now, obviously, you all are going into medical side, right, at some point. I am interested in the pox virus. I'm interested in adenovirus. I'm interested in polyomavirus. I'm interested in herpes virus. For example, I can say, how does this polyomavirus causes tumor? Do you understand? You should be able to answer that to me. That's a long answer question, okay? So this is, please stress on this. Now, positive stranded, we went straight on to poliovirus, yes or no, and coronavirus. What do they cause? What's so interesting? Everything interesting about them I've already put in the PowerPoint. Oh my God, okay. Oh my gosh, rabies virus, influenza virus. Okay, so rabies virus, influenza virus. One thing definitely I would be asking you is discuss. Why is it so hard to make a correct vaccine for the correct flu virus? Because it is changing. And where are the changes? Yes, but yes.
I've been studying. I have to do better on those tests. So,
darling, if you think you're stressed out, you're not even understanding how stressed out I am. Okay, so here is the double-stranded RNA virus, is the Rio virus. All right. And the Rio virus, so keep this in mind for the influenza virus, okay, that question. All right, just don't write genetic shift. You have to tell me what genetic shift is. And why was it so important? Remember the fragmented genome, okay. So just don't write one sentence because it's a long answer question, okay. All right, so, and if I'm very excited about something, that means it's on the test, okay. So don't make me say too many things, then it'll be cheating. So now viruses that, of course, we want to study this. Why? Because these are what is so interesting about retrovirus. Even though the retrovirus is single-stranded plus, right? There are two single-stranded which are plus. So there are two single-stranded, they're plus. Now initially the plus was directly used as an mRNA. Nah, we are not going to do this. The plus is DNA strand, obviously a minus DNA strand, okay, for one of them. The second one, again, the same thing is repeated. And then for the minus, you are going to use the DNA polymerase to make the second DNA strand. Then you are going to jam it into the host, and then finally things happen, okay? So this is interesting for us. Why it is doing this, okay? Why would it not just do that, okay? So this is very complicated, but as long as you know that tRNA is being used, simplify this. Directly go from here, primer, and directly go into here. You are going to synthesize this, and you're going to get rid of the RNA, because that single negative DNA, you are going to keep this just the primer, and you're going to complete and make it into a double strand, okay? If you have difficulty, ask me. We'll get out in a few minutes, ma'am. Hepadna virus. What is so interesting about Hepadna virus? Remember, here is my partial. It looks like a C surrounded by a circle. Viral ecology, I'm not interested. I'm definitely very interested in viriyoids. Viriyoids, prions are there. Differences, I can ask you to discuss. Viriyoids and prions. At the end, do you see these questions in here? I like these questions. I always look at these questions myself. Do you understand? Okay. And then for 11 is easy. 11, I only did this. I started from, oh, your book is different from mine? No. So genetic transfer, I did combination, complementation, transformation, competence, transduction, especially generalized and specialized transduction. What's the difference between this and the GTAs? Conjugation, almost done. and formation of HFR strands, and we are done. We are done with this. And then we are not going to talk about this. We are not talking about this. We are interested in the CRISPR. Please make sure that you know CRISPR well. I'm not telling you too much of this thing because it'll be cheating, but CRISPR is important. If I'm saying CRISPR is important, it is on your test. Now after this I cannot say, be careful, others run away with your pencil. Okay.